If you ask a plant what it's most grateful for, the answer is probably its leaves. A leaf's claim to fame is photosynthesis, using energy from the sun to convert carbon dioxide gas into sugars. In turn, those sugars feed the entire plant body down into the roots and help to build every single cell wall of the entire plant's body. Leaves also act as the plant's lungs, experts in gas exchange. They inhale carbon dioxide and exhale oxygen, so us humans are also grateful to leaves. In this episode, we'll be diving into leaf anatomy, how it's related to leaf function, and how it adapted to extreme environments. I'm Dr. Joyce Onyenedum, and I'm a botany professor at New York University. Welcome to Build a Plant, my video series where we discuss the hot topic of plant anatomy. Early land plants are thought to have been leafless organisms sporting only branching axes with terminal sporangia. Leaves then independently evolved, not once, but multiple times. These diverging evolutionary paths led to the diversity in leaf forms across lycophytes, ferns, and seed plants that we appreciate today. Leaf development is a fascinating process that begins at the shoot apical meristem. When a leaf primordium first forms at the peripheral zone of the meristem, it's spherical, but it soon flattens and differentiates into an adaxial or upper surface and an abaxial or lower surface. As the leaf develops, distinct tissue layers form. Let's take a look at a cross-section of a Ligustrum leaf. The outermost layer is the epidermis, which protects the leaf and is often coated with a thick layer of wax known as the cuticle. The epidermal layer also contains stomata, mouth-like openings for gas and water exchange that are surrounded by guard cells which regulate stomatal opening. Beneath the epidermis lies the mesophyll. These cells are packed with chloroplasts because this is where most of the photosynthesis happens. This layer is divided into palisade mesophyll at the adaxial surface with cells arranged like pillars to maximize light absorption, and spongy mesophyll at the abaxial surface with irregularly shaped cells that create air spaces for gas exchange. Running through the leaf are veins, vascular bundles that contain xylem for water transport and phloem for sugar transport. The arrangement of these veins can vary greatly between species, from the parallel veins of grasses to the intricate networks in broadleaf plants. Leaf anatomy dramatically differs between plants in dry and wet habitats, reflecting their adaptations to water availability. In dry ecosystems, plants exhibit xeromorphic traits to conserve water, exemplified here in the Deserts of the Americas gallery of the New York Botanical Gardens Enid Haupt Conservatory. These include smaller leaves that can be highly modified to be succulent or become spines. Their anatomy is adapted to prevent desiccation, such as thick cuticles, robust epidermis, sunken stomata, and abundant trichomes. In contrast, leaves in wet habitats are usually larger, with higher surface to volume ratios to maximize photosynthesis. They have thinner leaves with less dense mesophyll for efficient gas exchange, and higher stomatal density for enhanced transpiration and cooling. Leaves in aquatic habitats may have specialized tissues like parenchyma to provide buoyancy and gas exchange. These anatomical differences highlight how plants have evolved to optimize their leaf structure based on water availability in their environment. Another example of interesting leaf anatomy can be found in cam plants. These plants are adapted to arid environments and do a special form of photosynthesis compared to typical C3 plants. In order to do cam photosynthesis, these leaves are thick and have larger cells for storing water and acids, allowing them to keep their stomata closed during the hot day and to open them at night to take in carbon dioxide. Some of the most morphologically and anatomically extreme leaves can be found in carnivorous plants, which rely on highly specialized leaf structures to trap and digest animal prey. There are two main types of traps, active and passive. Active traps, like the Venus flytrap, use hinge leaves that snap shut when trigger hairs are touched. These trigger hairs are highly sensitive trichomes, modifications of the epidermal tissue layer that, when stimulated, send rapid signals which cause the leaf to close and trap the insect inside. Passive traps rely on more static mechanisms. Pitcher plants like Sarracenia and Nepenthes make leaves that form pitfall traps. The tops of leaf traps have epidermal modifications that make them slippery so that insects can't get a grip and fall in. The traps are filled with digestive fluids secreted by specialized glands to break down prey. Sundews use another type of passive trap. These leaves are covered in trichomes that are modified to be glandular and secrete a sticky mucilage. When an insect lands on the leaf, it gets stuck in the mucilage, and the glands secrete digestive enzymes to break down the prey. The anatomical modifications in these plants are what allow for their unique mode of nutrient acquisition. Let's recap. Leaves are incredibly diverse and multifunctional organs, serving not just as photosynthetic powerhouses, but also as sites for gas exchange and water regulation. Their intricate anatomy is adapted for these purposes, with distinct tissue layers working in harmony. We've also seen how some plants have taken leaf adaptation to the extreme, with can plants modifying their leaf anatomy for specialized photosynthesis, and carnivorous plants evolving intricate trapping mechanisms. In our next video, we're moving from vegetative to reproductive anatomy as we explore seeds and flowers.